Professor Hope Johnson speaking or sharing with us. And Hope is interested in environmental microbiology and geobiology. Her research focuses on manganese oxidation and cyanobacteria that form macrostructures. She teaches genetics and molecular biology, general microbiology, and advances in microbiology at Cal State Fullerton. Dr. Johnson holds a master and PhD from Stanford University, and her undergrad was at Cornell. Please welcome Dr. Hope Johnson.
Thank you. Thank you very much for having me here.
Can we hear?
Oh, can you hear me in the back? Yes. Okay. Okay, so since it's not a super big crowd, so if you have any questions during my talk, feel free to ask them. I'm happy to make sure everybody's on board instead of leave anybody behind. So what I'm going to be talking about today really grew out of a sabbatical I had a couple years ago at Caltech with Woody Fisher. So Woody Fisher is a geobiologist at Caltech, and we had some similar ideas. I've been studying manganese oxidation for a long time in bacteria, and I was interested in whether cyanobacteria could do this reaction. And Woody was interested in, primarily a geologist, but also a geobiologist, in understanding, the big events in Earth's history. And so that's where this project got started. So it's a collaboration with Woody Fisher's group at Caltech. So if we want to understand life on other planets and the possibility for life out there somewhere where we're looking, when we're looking in the night sky, one of the best ways to understand what might be possible out there is to just understand the history of our own planet and how life started here and what made life possible here. So that's, as geobiologists, that's one thing we're always looking at, is how life and Earth history interact. So there have been about 1,800 planets that have been described outside of our solar system. And so that's quite a few, 1,800, but not all of them are going to be hospitable to life. So the ones that are, we call those the Goldilocks planets, so the ones that are just right. And what that means by being just right is that it's not too hot and it's not too cold. So if it's too hot, chemistry takes over and life doesn't really have a chance. If it's too cold, the reactions are so slow that it would be really hard for life to take advantage of the chemistry that's required for energy and having life. So of those about approximately 1,800 planets that have been described, only 16 are in this, what we consider the habitable zone, where it's the right temperature for life. And clearly, Earth is one of those planets because we have life here. So in addition to temperature, the size of the planet matters. So if it's too small, it won't be able to hold an atmosphere. But if it's too big, that atmosphere will just be oppressive. So the right size and the right temperature is important when we're looking for planets out there that might have life. So besides the size and location of the planet, the other things that we need to consider are the essential elements. So we know what those essential elements are on Earth: carbon, nitrogen, oxygen, hydrogen, phosphorus, sulfur. We know what those things are. That doesn't mean these are going to be the same elements that are required for life somewhere else, but they give us a starting point. We also know that water is important, because most reactions take place in water. As far as we know, life would require some type of liquid to exist in. Did you have a question?
Yeah.
Yeah.
So are we speaking about or referring to life as life on planet Earth as we know it? Or are we considering other types of life forms?
So, and I'm talking about life, we're using Earth as our model, but also what could we look for somewhere else? So if we're looking for someplace else, it has to be the right temperature, it has to be the right size of planet, it has to have the elements that are essential for life. So things that we know that are probably required, that we know are required, that might be something else, but if they have the elements that we know are required, that makes it easier for us. There has to be water, some kind of liquid for life to exist in. And there has to be energy. And so this is true of life on our planet as well. If we go somewhere, as long as we have these essentials, there's the possibility for life. So having these things doesn't guarantee life, but it increases the probability of it. Okay, so if we go to some exotic location on our planet, one of the places people are interested in looking for life right now is in the deep biosphere, so miles down into the planet. And seeing what is down there, the main question that comes up is what is down there that can be serve as an energy source. So energy is really important. And as an environmental microbiologist, we think about energy a lot because the microbes are so diverse. They usually can get energy from any reaction that's exergonic. So they can harness that energy. So when we're thinking about life of microbes on this planet or somewhere else, we are always thinking about energy. Okay, so if we think about how life started on this planet, we think that it has to have the energy, essential elements, and water. So a lot of people look to hydrothermal vents as a place where life could have started because it has all of those elements. It has the elements of life coming up through the hydrothermal fluids, and then water, and then lots of energy in these hydrothermal fluids. So now I'm going to talk just about our planet and the main events that are important in our Earth history. Earth, about 4.5 billion years ago, we know the age of the Earth, not because of the rocks that are on our planet. We don't have any rocks on our planet that have been discovered that are that old, but from meteorites. So from looking at meteorites, we can date the age of the Earth. And we do that using uranium to lead radioactivity. Or not, radiometric dating, not radioactivity, radiometric dating. Okay, so the next big event is about 1 billion years later, and that's when there's evidence for life. So life may have existed prior to that, but we don't have any evidence of that. So the evidence that we have is around 3.5 to 3.8 billion years ago. So I'm going to tell you about that evidence. There's 3 lines of evidence that dates life to that time on our planet. And so this is the kind of evidence that we would also look for on another planet if we were interested in life on another planet. So if you want to pass that around, this is a stromatolite. So one of the lines of evidence are these rocks called stromatolites. So these are stromatolites, they have a layered structure. So there's a layered structure that start from a point of accretion. Basically, they make these layered mounds, and they're thought to be caused by microbes making a sticky biofilm that traps and binds sediment. And so you get this sedimentary layering. So stromatolites can be very old, or this is a modern stromatolite in Australia in a place called Shark Bay. And so you can find these modern stromatolites, although they're not very abundant in today's world. They're not very abundant in today's world, but based on the geologic record, they used to be pretty abundant throughout time until about 500 million years ago. So these stromatolites have been dated to about 3.7 billion years ago. Okay, so they can form abiotically, but the shape of them, a lot of the shapes of them, there's no abiotic mechanism that's known. So it's thought microbes would be involved in that. Another line of evidence for life at 3.5 to 3.8 billion years has to do with carbon isotopes. So carbon can exist as carbon-12, carbon-13, and carbon-14. And most people are familiar with carbon-12 and radioactive carbon-14. But when we're talking about trying to date things that are really old, carbon-14 is going to be gone. So there's going to be no carbon-14 because it has a half-life of only around 5,700 years. So if we're looking at billions of years ago, there's no carbon-14 left. But there's also a stable isotope, carbon-13. So most of the carbon on our planet is carbon-12, but there's a little bit of carbon-13, a small fraction of carbon-13. And what we know about life, about the enzymes that have evolved on this planet, in all the organisms studied is that organisms, life, likes light carbon. It preferentially uses carbon-12. So if there's a pool of carbon-12 and carbon-13 out there, so let's say 90% carbon-12, 10% carbon-13, life will preferentially take up the carbon-12. So that means that life is going to be much lighter than just things that are formed abiotically because it likes this light carbon. So enzymes have evolved to really use light carbon, the carbon-12 instead of carbon-13. It's a little bit more difficult to break a carbon-13 bond, so that's probably why life likes carbon-12 better. But also these enzymes have evolved to be optimized for carbon-12 because that's the most abundant. So we can go out and determine the fraction of carbon-12 to carbon-13 that's in anything. We can go out and look at it in the soil. We can look at the carbon in the air. We can see what fraction is carbon-12 versus carbon-13. And that's been done with rocks from all different ages. And some rocks from Greenland that are 3.85 billion years ago, they've shown that they're enriched in carbon-12. So that means that life must have been involved in enriching that carbon to carbon-12. Otherwise, you wouldn't see any enrichment of carbon-12. Okay, so we have stromatolites. We have carbon-12 enrichment, because life likes carbon-12. And we have microfossils. So microfossils are just like regular fossils, but you have to look at them under the microscope because they're really small. And they're not super compelling. It's not like looking at a fern fossil. That's pretty cool and beautiful. These are just little round shapes, sometimes spheres. And the idea is they look microbial. So as a microbiologist, I find this the least compelling evidence because I can look at a sample from some sediment and I have a hard time figuring out what are cells and what are not cells just by looking at it. But when this is done, the one exception to that I would say are these filamentous microbes. So filamentous microbes are easy to pick out. And so here we see some filamentous microfossils. So microbes that form filaments, here they are with less magnification. And this sample in particular, it has been also, they've looked at the carbon isotopes. So it's a combination of just the microfossil and showing the carbon isotopes that a carbon-12 is enriched in these samples. So these are dated to around 3.4 billion years ago. Yes?
Are these mostly found in frozen type areas?
So the question is, are these found in frozen type areas? No, not necessarily. So they're found in places where the rocks have not been weathered very much. So that means lots of times Australia. People go to Australia to look at really old rocks where the rocks haven't weathered. So when there's water, rocks get weathered. We know when it's dry and not so much water, they can be preserved longer. So some of the oldest rocks are in Australia. That doesn't mean there's not rocks that are really old under ice that we haven't discovered yet. Okay, so life's significant event in Earth's history. The next most important event, and one of the ones that changed our planet so much that we are here today, that allows us to be here, would be the oxygenic photosynthesis. So the evolution of oxygenic photosynthesis by cyanobacteria. And what is important about that is prior to this, Earth was very reducing. It didn't have any oxygen. It had only reduced compounds, so like only iron 2, no iron 3. There wasn't much nitrate, not much sulfate, things that are very abundant today. Those chemical compounds would not have been present on early Earth because there was no strong oxidizing agent. So the evolution of oxygenic photosynthesis in cyanobacteria has been dated to about 2.7 billion years ago, and that is based on molecular clock data. So basically assuming some mutation rate. And then we have evidence that around 2.3 to 2.4 billion years ago was a great oxidation event. And that is when oxygen started to accumulate in the atmosphere to 1 part per million, which is still pretty small. Present atmospheric levels are 200,000 parts per million. So this is not very much oxygen, but it's enough oxygen that it basically changed our planet completely. So then our planet now became oxidizing, and it really changed the rocks that are there and what life was possible. Okay, so we know, well, this is a timeline of the age of Earth, so present level, and then down back to 4 billion years ago. And this is the level of oxygen, so present atmospheric level, and then just in atmospheres. And you can see there's no oxygen. It's thought that oxygenic photosynthesis arose here at some point. These are what are thought to be whiffs of oxygen. So in some environments, there would be some amount of oxygen formed. And then you have the great oxidation event here that caused the oxygen levels to significantly increase. And then that was relatively stable for a while until then you get up to present day atmospheric levels of oxygen. And so how do we know that there was no oxygen here and then there was oxygen here all of a sudden and then here? It has to do with minerals. So we have what are called proxies for oxygen. So I'll talk about one, which is pyrite. So iron sulfide. Iron sulfide is not present if there's molecular oxygen. So molecular oxygen will oxidize iron to iron 3, so you don't find pyrite. There's a molybdenum, so molybdenum does not become oxidized in the absence of oxygen. Manganese is thought to not be oxidized in the absence of oxygen, and I'll talk about that. So you can look at these, and there's the sulfur isotopes that are also used. So there's a bunch of different techniques, and I'm not going to go into the details of those techniques, but there's a bunch of different techniques that are used to date oxygen, and that's based on minerals pretty much. So looking at the minerals that are there, and we know that those minerals can't form in the absence of oxygen. But the one I'll talk about more is pyrite, because I'm going to show you some sedimentary data. Okay, the implications of the Great Oxidation Event were spectacular on Earth. This is arguably the biggest change that happened on Earth since it was formed or life arose. One is that prior to this, there was no oxygen in the atmosphere. So oxygen is bad. It oxidizes things. We take antioxidants to help prevent the bad effects of oxygen. Prior to that, there would be no reason for life to have any of these oxygen antioxidant strategies. And so a lot of life would have died in the presence of oxygen. There was also no ozone level, no ozone in the atmosphere. So that means that radiation would have been significant, and most of life could not live on the surface. So life had to exist below the surface because it was just too dangerous to be on the surface. So oxygen being present led to new metabolic pathways. And these new metabolic pathways arose because, one, there's more energy, but also there's all kinds of new compounds around that weren't abundant before. So things like sulfate and nitrate, they were not, and oxidized iron, they were not present prior to the GOE. The other thing about oxygen being present is that oxygen is a great electron acceptor. So we breathe with oxygen. Oxygen is our electron acceptor. And breathing with oxygen gives us a ton of energy. And being able to get a ton of energy means that there can be lots of diversification. There's energy to spare. You can carry around genes that don't necessarily work very well. And then eventually maybe they'll have a purpose. It also leads to multicellularity. So there's all this energy that's now available from having oxygen to be able to breathe with oxygen instead of some other compound. Okay, so cyanobacteria, you may know them as blue-green algae. So they are the bacterial form of the oxygenic phototropes. They're responsible for about 50% of the primary productivity in the oceans. The other 50% is eukaryotic algae. They produce oxygen through photosynthesis. So they produce 50% of the microbial oxygen. So microbial oxygen makes oxygen production is 50% of the total oxygen production. So we can think about plants doing 50%, microbes doing the other 50% of oxygen production on our planet. The cyanobacteria, they share an ancestor with chloroplasts, so they're clearly related to chloroplasts. That's been shown clearly. It's thought that they were probably responsible for the formation of stromatolites. There's not a lot of good evidence for that. It's just more of an intellectual, like these organisms, they're important for stromatolites today. They were probably likely important for the formation of stromatolites a long time ago. And then these organisms are responsible for producing the great oxidation event. So these cyanobacteria hold a special place in terms of Earth history.
What's a chloroplast?
Oh, a chloroplast is the organelle in plants that does photosynthesis. Okay, so now I'm going to start talking about oxygenic photosynthesis because that's the process that produces oxygen. And so if this is our cyanobacteria, what cyanobacteria do to grow, and this is what plants do too, is they take water and they oxidize it through light to produce molecular oxygen. And they take CO2 from the atmosphere and produce cellular material. And so this is often how people think of oxygenic photosynthesis, that these two processes happen together. And often they do happen together. In plants, they're totally linked. In microbes, they're not always linked. So I want to separate these two processes and talk about this part as the photosynthesis, the part where water is oxidized to make molecular oxygen. And then this part where CO2 is used to make cellular material, we call that carbon fixation. These two processes are linked by an electron, by electron flow. But this is the part I want to tell you more about today. So I'm not going to talk about this part anymore. Okay, so the enzyme that does this, the enzyme, it's a complex. It's called photosystem 2. And what it does is it takes electrons from water. So my lab, we talk about electrons a lot, and I know they're kind of abstract. So if you have any questions, please feel free to interrupt me, because not everyone thinks about electrons all the time like we do. So it takes electrons from water and produces molecular oxygen, and then those electrons are transported, are moved away. And so those electrons then would go through an electron transport chain. And then they can be used to make cellular material. Okay, so I'm going to simplify this even a little bit more. And so chlorophyll holds an electron, and chlorophyll is special. Because if a photon of light hits chlorophyll when it has an electron there, what happens is the energy of that electron changes. When light hits it, when it's in the chlorophyll, it gets more energy. So its energetic state changes. So we call this, so this is a high energy electron, and we say it has more negative potential. So more negative electrons have more energy. And it started out with a low energy, as a low energy electron with a more positive potential. So we have light hitting an electron, and then that electron changes potential, and then that electron can go down through an electron transport chain. It makes ATP and can be used for carbon fixation. So basically, we're just moving electrons around. And the beauty of photosynthesis is taking light and changing the potential of that electron. Because if we think about how we get energy, how all energy happens in the cell pretty much is through moving electrons around. That's where all energy comes from. And this special form, photosynthesis way of making energy, is changing the energy of an electron so that it can move around in a different way that benefits us and gives us ATP.
Did I miss ATP with it?
Yes, so if we have electrons and they move down an electron transport chain, that results in ATP being formed. So, what is ATP? ATP is the energy of the cell. So if we are thinking about how energy is moved around in a cell, the molecule that does that is that energy, is ATP.
say the transport chain, what does that look like? I mean,
So an electron transport chain is in the membrane, in the cell membrane, and it's a series of proteins in which electrons get transferred through. And that's how we get energy. That's how the cell makes energy.
ATP stuff?
Yeah, that's how it makes ATP, by transferring those electrons from one protein to another.
All right.
Okay, so to simplify this even more, once that electron gets its new potential, so it gets a lot more energy and is moved away, now you have a chlorophyll that needs an electron. So it just donated that electron away. It's changed its potential. It's moved down the electron transport chain. Maybe it went to carbon dioxide fixation. And now it needs an electron. And that electron comes from water when it's oxygenic photosynthesis. So when oxygen is being produced, that electron comes from water. And we know that currently microbes are out there right now that can do this same process, but their electron comes from hydrogen sulfide or iron, organic compounds, something besides water. So we know this, and that's called an oxygenic photosynthesis because oxygen is not produced. But it's doing the same thing, it's just getting its electron from somewhere else. And this is not called a photosystem 2, it's just called a reaction center, and these reaction centers have different types. Okay, so I'm going to be talking about electrons now and try to show you what is this, the super awesomeness of photosystem 2 and what it can do with electrons. So this is the potential. So the more energy you have, the more positive of the electron. And there are these different photosystems. There's photosystem 1, reaction center 2, reaction center 1. So these are in photosystem 2. So those are the reaction centers we have on our planet right now that microorganisms can hold. And one thing I want you to notice, if you look at all these reaction centers, their energy is different. The type of electrons that they can accept is different than photosystem 2, which gets its electrons from water. Okay, so water does not like to give up its electrons at all. It's a very difficult process getting those electrons off of water and making molecular oxygen. And these photosystems, they can't do it. They just don't have the right potential to pull those electrons off. Because electrons are always pulled from something more negative to something more positive. Okay, so this photosystem 2 is special. But all of these photosystems are related to each other. They all have a common ancestor. If we just look at 2 of these, with the cyanobacteria, this would be photosystem 2, and this would be in the purple bacteria. They're related to each other. They share a common ancestor. So all of these photosystems, except for photosystem 2, they all do these reactions that are basically low potential. They aren't at a high enough potential to take an electron from water. So whatever that ancestral photosystem was that led to these different photosystems that we have today, we think that it probably did low potential reactions. Because of the photosystems today that are in this nice phylogenetic tree, the photosystem 2 is the only one that can do a high potential reaction. So we think the ancestral reaction center was probably low potential. So it could do things, use hydrogen sulfide as its electron donor instead of water. So it couldn't do any water oxidation. And here today, we have our photosystem 2. It can take electrons from water and produce oxygen, and that's our oxygenic photosynthesis. But if we think about evolution as a stepwise process where changes are made and then they're beneficial and then they can, through natural selection, they last, if we think about what that step might be here, we think it might be a photosystem that had a high potential reaction. So before it could take electrons from water, which is kind of like a very difficult reaction leading to toxic products, there was probably a high potential reaction for photosynthesis somewhere in there, and that's where we think a manganese photosystem would be. So we think a high potential photosystem would have existed as an intermediate and be this missing link between the ancestor and photosystem 2. Because it's not clear otherwise how you would get something, how this photosystem 2 that can do this really cool reaction that most of life depends on our planet right now, how that could have evolved without going through some type of intermediate step. And we think manganese is the key to that. So here's just a little bit to give you a more cartoon-like view of what these look like. They all look very similar. And so this is the one we have today with this water oxidizing complex where you take water and produce molecular oxygen. This would be some ancestor, and we are proposing something in the middle here happened. So how are we going to show this? We're going to show this by looking at what is modern today and seeing whether it can do this.
Man, is that magnesium?
Manganese.
Manganese.
Manganese. Yeah, Mn is manganese. Okay, so we're going to go through the rationale why we think this is a good hypothesis and why we want to test it. So these photosystems, they all have very similar structures, and so that also supports that they came from a common ancestor. But photosystem 2 has this unique part of its structure called the water oxidizing complex. And this water oxidizing complex is where the electron goes from water. That's its first stop before it goes to the chlorophyll.
Goes to the what?
Chlorophyll.
Chlorophyll.
The chlorophyll. So this water oxidizing complex, it has manganese. So it has manganese. It's a complex of these 4 manganese metals and the calcium and some oxygens, and then there's some waters attached. So in order for photosystem to work today, it has to form this complex in order to get electrons from water. And when it forms this complex, it basically is oxidizing manganese. So as this complex is formed, so these manganese are like +3 and +4. Manganese 2 that the enzyme uses, it takes that and it actually removes electrons from these manganese to form this complex. So we've got manganese, which is important in terms of water oxidation. Okay, so that's our biochemical and structural information that points us in this direction. We also have a geological rationale for this. And as I mentioned before, manganese oxidation can be a proxy for the presence of oxygen. Because we're proposing here that it could be photosynthesis, but prior to, right now, most people think it's just molecular oxygen that can oxidize manganese. And the organisms, I've studied manganese oxidation in bacteria for many years. All of those reactions, they require oxygen. So that has always been thought of as a key when you find manganese insoluble manganese in the sedimentary record, that's an indicator that oxygen was there. Because you don't get insoluble oxides unless you have oxygen. But now we're proposing that maybe this could be photosynthesis. So I'm going to show you some geological data, some sedimentary data from my collaborator. But first I want to explain how manganese accumulates in the sediment. So when manganese is oxidized, so we have manganese 2, which is soluble in water. When it becomes oxidized, it becomes insoluble. And that means it's going to precipitate down and it'll go to the sediment. And that, over geologic time, gets reduced and turned into a manganese carbonate. So people are looking for manganese carbonates in the rock. So if you see any manganese accumulates in the sediment, that's a sign that this happened here, that you had some oxidation. So this is a sedimentary data from my colleague and his group at Caltech. And they went to South Africa and they got some drill cores, and so they could look at the sediment that's really old. So this is 2.4 billion year old sediment. And one thing that they see is that there's lots of manganese. So this is weight percent of manganese. You can see you get a lot of manganese here. These are 2 different cores from the same location. You get a lot of manganese here. So you have an abundance of manganese. So that suggests that manganese must have been oxidized and insoluble for you to find it in the sediment. The other thing that you see is you see pyrite. So we see these high levels of manganese, and you also see pyrite. And pyrite is not going to accumulate in the presence of oxygen. So if there's oxygen, there won't be pyrite. We think of them as being exclusive. So when we see pyrite in the sediment, in old sediment, that's an indication that there was no oxygen. So these sedimentary samples, so this is like depth here along the sediment core, and then we see manganese, and then these little cartoons here indicate where you see iron sulfides, which is the pyrite. So what this indicates is that there was oxidized manganese, but there wasn't molecular oxygen.
Did there have to be a lot of manganese all over the earth to have this happen?
So the question is, does there have to be a lot of manganese all over the earth for this to happen? And you would have to have higher levels of manganese, and the early oceans are thought to have had high levels of manganese.
We don't have that now.
We don't have that now.
Who ate it?
It sedimented out. So we don't have high levels of manganese in our oceans today. But about 70% of our ocean is covered with manganese nodules, which accumulate lots of other metals. So we have a lot, most of the ocean is covered in some manganese rocks, the ocean floor.
Not very handy.
No, kind of hard to get at. Okay, so that was our biochemical structural data, or rationale, and the geologic rationale. And now I want to talk about the physiological rationale for why this would have been an intermediate process. And that is because, as I mentioned, oxygen is dangerous. Molecular oxygen reacts with DNA and proteins and membranes, and it produces superoxide, hydrogen peroxide, and hydroxyl radicals. And these, we know, are all bad. And we have enzymes, and every organism that lives under oxygen conditions has enzymes to basically take care of these toxic products. So if we are thinking about when oxygenic photosynthesis evolved, these organisms would not have any of these enzymes that could help ameliorate these effects of these toxic products that would inevitably form if oxygen was made. So how could these cells have survived making a toxic compound that could potentially kill them? Well, it turns out that manganese, if the cells accumulated manganese, if they oxidized manganese so that there was manganese around them accumulating, that could act as an antioxidant. Because we know now that there are that manganese can complex different types of small molecules and act as a superoxide dismutase, a catalase, or react with hydroxyl radicals. So manganese acts as a good antioxidant. So if the cells were accumulating manganese and then they started to produce oxygen, they would be able to more likely to survive than if they didn't. Okay, so we know organisms now that accumulate manganese as a defense against oxygenic stress. Okay, so this missing link, this organism that could potentially be holding this intermediate photosystem doesn't exist probably today. So we can't go back in time and we can't go and look for an organism that can do that. But what we can do is we can look at modern organisms and modern photosystem 2 and see if we can manipulate the system to see that it oxidizes manganese. Because if it does do phototrophic manganese oxidation now, that would provide evidence that it may have been done in the past. We are working with Cyanobacteria, Synecocystis, species PCC 6803. This is what it looks like under the microscope here. It makes these green little spheres, and this is an electron micrograph of it. These lines here are its thylakoid membranes where it does photosynthesis. So this is our organism, and we are working with this organism because it's been used to study photosystem 2 for many, many years. Since the 50s, people have been using this organism to understand photosystem 2. So one of the tools that we'll have is mutants, which we have started working with. Okay, so this is some work from a student in Woody Fisher's lab, Usha, and she has shown very nicely here that this cyanobacteria can oxidize manganese. So if we fiddle with the system a little bit, we can get it to oxidize manganese. So this is a manganese that's oxidized. This is time. We see that manganese accumulates, oxidized manganese accumulates over time with the cells. And we see also that this reaction is light dependent. So if we are talking about a photosystem that's going to oxidize manganese, it has to happen in the light and not in the dark. And so here is in green here in the dark. We see nothing happens in the dark. So it's a light dependent reaction. We can also add this herbicide called DCMU that inhibits photosystem 2. It's a common herbicide that's used. And we see that when we add that, so that's the blue, we don't get any manganese oxidation. So we know it's a light-dependent reaction, consistent with it being photosynthesis. It's inhibited by a known inhibitor of photosynthesis. And when we look at superoxide dismutase and catalase, which are enzymes that could inhibit other pathways for manganese oxidation that we already know. We know there's other pathways for manganese oxidation, and we can inhibit them with these enzymes, and it's not inhibited here. So right now, everything is consistent with these cyanobacteria are able to oxidize manganese. So modern cyanobacteria that can do that. So, I mean, we were very excited about this, but we wanted to look at it more in a more reductionist kind of way to make sure, like, you know, cells, there's lots of things going on with cells. Cells are complicated. And so we wanted to see whether photosystem 2 itself could oxidize manganese. And so here's a little cartoon of photosystem 2. When light hits it, we already know that it can do this. Take water and make oxygen. And here's an electron acceptor to just get those electrons from the other side. And so our question is, can we give it manganese, and will it make manganese 3? Will it oxidize manganese? Okay, and it does. It does oxidize manganese, so that's pretty exciting. So this is just a colorimetric assay that's used to measure oxidized manganese. Here we can see that we're getting a lot of oxidized manganese. Not so much in the dark, and under abiotic conditions we get a little bit of manganese oxidation. So when we put these compounds in the light, the DCBQ is actually a colored compound. Colored compounds in the light, they do things that are often unexpected. So that's probably why we have some abiotic reaction right here. But it's much less than our biotic reaction. So we think that we are getting photosystem 2 clearly oxidizing manganese. Another line of evidence that we can do to pull this together is if we have this reaction going on, so we have water and water, electrons are being taken from water to produce molecular oxygen, and we add manganese in here. Manganese would be a competitor with water. So these two substrates, these are both substrates potential of this enzyme. If we add them both in, they're going to be competing. And under that situation, we would expect less oxygen to be formed. Because sometimes manganese is going to get oxidized instead of water. So they're competing. And so what we can do is we can measure the oxygen that's formed. Here's no manganese. We see we get some oxygen formed. We can also measure the amount of electrons that went through the system, through photosystem 2. We have some electrons here. When we add manganese, we get way less oxygen formed, and we actually get more electrons going through. So that's very consistent with manganese being used as a substrate for photosystem 2. Okay, so we're very excited about this because we can show we have some preliminary evidence here that this is happening in these modern day cyanobacteria, which would be very consistent with this being an intermediate step. And so we wanted to know how widespread this was. And so we looked at some eukaryotic algae. And one of my students, he was able to isolate some eukaryotic algae and then tested it for manganese oxidation. And we see that it also oxidizes manganese very slowly, but it does. It's inhibited in the dark, and it's inhibited with a herbicide. So when we inhibit the enzyme, we don't see that. But when it's in the light, we do see that it oxidizes manganese. So this is exciting because it expands basically the number of organisms where this is potentially possible and the environments that this could be happening in. Okay, so what does all this mean? This idea I'm showing you some preliminary data here about that we do think that manganese phototrophy takes place now in cyanobacteria and eukaryotic algae and by photosystem 2. So this supports a role for ancient phototrophic manganese oxidation. So it's consistent with being the missing link in the evolution of photosynthesis. We also have figured out that manganese oxides are no longer a good proxy for oxygen. So if manganese can be oxidized in the absence of oxygen by light when oxygen is not present, then that means that manganese oxides don't need oxygen to form. So we wouldn't want to use that as an indicator of whether there was oxygen or not in the past. Okay, so we don't want to use this as a proxy for oxygen in the past anymore because it could occur by photosynthesis, which doesn't require oxygen. Okay, it also changes our view of manganese oxidation. So manganese oxidation is a microbial process. It's really important in the biochemical cycling of the elements on our planet. It affects iron cycling, it affects sulfur cycling, it affects carbon cycling. So these organisms that can oxidize manganese play this important role. And now we're showing that there's new organisms that can do this, new mechanisms for how this process can happen, and that manganese oxidation might be way more prevalent than we had ever thought. So if all these phototrophs are able to also oxidize manganese, and there's phototrophs everywhere, this really means that there's a lot of manganese cycling on our planet. And that has implications for nutrient cycling and trace metal cycling, and even cycling of things we care a lot about these days, so it's just carbon cycling. But it also points to one of these new ideas in environmental micro that I find really compelling are about cryptic cycles.
What do you mean by cryptic?
Cryptic meaning that we can't really measure the products of the cycle. So in general, when we look for cycling, we look for the substrate and the product. And we look for some process that's going to accumulate the product. But manganese, when it's oxidized, is very reactive. So it could get reduced back to manganese 2 really easily. We already know this happens. And so there's likely, when there's small amounts of manganese, if all these phototrophs are doing this reaction or oxidizing manganese, there's likely a cryptic cycle. And that cryptic cycling can affect this nutrient cycling, carbon cycling, because the manganese that's produced is super reactive. So manganese is the most oxidized manganese is the most oxidizing compound behind oxygen in the environment. So we've got this strong oxidizer that could be potentially being made all the time, potentially oxidizing carbon, metals, all kinds of things. Okay, so with that, I want to thank Woody and his lab at Caltech. Because this really came out of our conversations and the time I had on my sabbatical. NSF and the CSUF Incentive Fund for supporting this research. And my lab here, so this is our recent lab lunch. And two people I want to point out that did the work. Joshua Phelan, he's an undergrad who did the work on the eukaryotic algae. And Leslie Kane-Barnice, who is a faculty member at Concordia University. And she did a sabbatical in my lab. And so she was really important in helping us get some of these experiments up and running. So if anyone has any additional questions, I'm more than happy to answer them. Thank you.
Could you go back one slide? So could you give us an example of the practical use for the research that's continuing?
So a practical use, that's a hard question. But we do know when we look at other manganese oxidizing organisms, not cyanobacteria, they can be used for bioremediation. because manganese oxides are highly reactive, so they can oxidize toxins. So we know they can oxidize like atrazine, they can oxidize uranium, so lots of different compounds. So there's a potential for bioremediation. With these cyanobacteria, I'm not sure that that potential is there. This is more of a theoretical direction. Any other questions? Thank you very much.